and welcome to this international symposium on food security and global health. I am Matthew Abraham, Managing Director and CEO of Kanandevan Hills Plantations Company Private Limited, KDHP, which is an associate company of Tata Consumer Products Limited and is the largest tea plantation company in South India, with an annual production of 25 million kilograms of various types of teas like orthodox, CTC, green, oolong, and specialty teas like rose tea and white teas. Before I introduce our speakers, and with the permission of a collaborating partner, President of Molecular Medicine Institute, Boston, USA, Professor Banu P. Jana, I would like to dedicate this symposium in honor of late Mr. Ratan Tata, Chairman Emeritus, Tata Sons, who was a visionary, a philanthropist, a humanist, and a globally acclaimed business leader par excellence. Quite a few of us at KDHP had the good fortune to meet him and interact with him during his visits to Munnar. Mr. Tata's legacy will continue to lead us on. I would be failing in my duty if I did not acknowledge the unstinted support we have received during the formation of this JV with Nytroponix LLC from the top leadership of Tata Consumer Products Limited. And our thanks are due to Mr. Sunil D'Souza, MD and CEO, Mr. Ajit Krishna Kumar, Chief Operating Officer, Mr. LKK, the previous Group CFO, and Mr. Rishi Gautam, Head Legal of TCPL. With this symposium, KDHP would like to showcase its partnership and commitment with the Molecular Medicine Institute and Nytroponix, a plant-based biotech company in Boston, a product of the MMI, to help launch the next green revolution and design new tea-based natural products for a healthier environment and society. Professor Jana, President and Director of the MMI, and the George E. Palade University Professor and Distinguished Professor, is central to this program. Before I invite him to chair the symposium, allow me to say a few words about his contributions to science and society. An adult human being is made up of approximately 38 trillion cells that communicate with each other via secretion to establish homeostasis and sustain life. Secretion is highly regulated, fundamental cellular process in living organisms, from yeast to the cells in humans. Neurotransmission for thought and memory, insulin release for maintaining glucose homeostasis, or endocrine pancreatic release to help digest food are a few examples governed by cell secretion. Nearly 30 years ago, Professor Jana discovered a new cellular structure called the porosome, a molecular nanomachine at the cell plasma membrane that enables cells to secrete in a precise and highly regulated manner. This discovery of the porosome, the universal secretory portal at the cell plasma membrane, as well as the subsequent dissection of its structure, dynamics, regulation, and composition, has resulted in a paradigm shift in our understanding of regulated secretion. Many diseases, including Alzheimer's, diabetes, cystic fibrosis, are now beginning to be addressed through this lens as a result of his discoveries. The discovery of the porosome has provided entry into a new era in drug design and development. For example, the recent studies demonstrate that a number of proteins in the neuronal porosome is altered in Alzheimer's. The introduction of new neuronal porosomes into Alzheimer's nerves is able to overcome the disease. Similarly, functional porosomes introduced into defective lung epithelia rescues from cystic fibrosis. Thus, Professor Jana's pioneering discovery of the porosome, the molecular understanding of its structure, composition, and function, and its translation to treat a wide range of debilitating disease, has brought great benefit to science and society. We are proud to have him here today with us and to chair the symposium. Welcome, Professor Jana.
Thank you. Thank you, Mr. Matthews, for this very generous and kind introduction. I couldn't have said about my own work better than what he did. Thank you. Well, good morning. Good morning, and again, my warm welcome to you all here in this beautiful Conn Hills Tea Estate, KDHP, in Munnar, Kerala, India, which is the world's highest elevation tea plantation at 2,400 meters, which is 2.4 kilometers. And Mount Everest is, I think, 8 point something, so you guys are getting very close. It's a beautiful place. KDHP is an associate company, as Mr. Matthew pointed out, with Tata Consumer Products, with research collaborations and partnership with our Boston-based Comprehensive Institute. It's an international institute, and we welcome anyone and every institutions to participate. And we have many, many participations in that institute, including the Karolinska Institute and other faculty from Harvard, MIT, and other institutions. And also the partnership here with KDHP with Nytroponix, one of our spin-offs companies that have been developed through the research that has been ongoing at MMI as a lead plant-based biotechnology company, dedicated in providing food security and health benefits globally. That's our mission of Nytroponix. So thanks to all of you for taking the time from your very busy schedule. I know Mr. Matthews was saying this. You guys are, many of you are in administration and in very different aspects of in tea production and marketing and so on and so forth and development. And you're busy people, so thank you for attending. And both people who are attending here as well as online. And as I share with you the progress made on various scientific fronts by the MMI and KDHP partnership, in the KDHP Nytroponix joint venture, I wish to remind you that the MMI was really established out of necessity to help translate new discoveries and inventions to benefit society. Otherwise, it always stays in textbooks. For example, this, as Mr. Matthew had pointed out, the discovery of the ozone. I'm a scientist, I'm not a businessman. And so I was helped by business leaders globally to start a new platform for proposal therapeutics, which now is helping treat many diseases. And I never dreamt in my life that the work that I do is going to be translated to help society in the health sector. So I'm very grateful to that for that. And similarly, with new understanding of the chromatin biology, and you'll hear today, the Nytroponix and KDHP is utilizing this new chromatin biology technology for novel tool and developments in the field that would allow us to understand not only and implement it for drug development and therapy, but also engineer plants that will be able to optimally capture carbon and nitrogen from the atmosphere to drawing up on these technologies for the third green revolution in the world. And as you all know, the first green revolution was the Bose technique of trapping nitrogen on a solid state surface catalysis, and then nitrogen fertilizer were developed, and then normal Bulwark wheat with the second green revolution. And I think we are really happy to report that we are in the verge of now taking forward the third great revolution here in India, here in Munnar, here in KDHP. So very proud to participate in that. And this symposium is to celebrate our new understanding of plant cells and to translate some of the understanding to benefit society. So in today's symposium on global food security and health, you'll hear lectures by 4 leading scientists who will enlighten us on the use of science to tackle some of the major issues facing food security and global health. And as you shall see in today's talks, India's national drink, tea, takes center stage. So with this, I would like now to give the podium back to my friend, the CEO, Mr. Matthew Abrahams, KDHP, to introduce our speakers. Mr. Abrahams.
Thank you, Professor Jena. Although our four speakers need no introduction, allow me to introduce them to you before I give them the floor. Our first speaker, Dr. Siddhartha G. Jena, is a cell biologist, biophysicist, bioengineer, and a postdoctoral fellow at the Harvard Stem Cell Biology and the Board Institute. He received his bachelor's from Harvard, majoring in chemistry, physics, and mathematics, and a minor in molecular biology, with Professor Martin Karplus, the 2013 Nobel laureate in chemistry, serving as his senior thesis advisor, and a summer research publication with Professor Jack Szostak, the 2009 Nobel laureate in physiology and medicine. Dr. Siddhartha Jena received his PhD from the Department of Molecular Biology at Princeton University. He works on the design principles of gene regulation and is broadly focused on teasing apart the organizational rules that govern genes. Today he will discuss the tools and approaches that could help launch the next green revolution. Our second speaker is Professor Robert Flomenoff, who is a professor in the Department of Medicine at Harvard Medical School and is chief of the Division of Hemostasis and Thrombosis at the BIBMC. His laboratory focuses on platelet and endothelial biology and mechanisms of thrombus formation. Professor Flomenoff has made seminal contributions to our understanding of the porosome, secretory machinery in platelets, called the open canaliculi system. His laboratory is also at the forefront of research, both basic and clinical, on the role of various flavonoids on cardiovascular health and well-being, which he will be discussing today, especially in the context of T flavonoids. Our third speaker is Professor Lisa Nottingham, who is Associate Professor in the Department of Medicine at Harvard Medical School and is Clinical Director of the Neuroendocrine and Pituitary Tumor Clinical Center at the Massachusetts General Hospital. In addition, she is the Director of MGH International Education Programs for the MGH Endocrine Division and of the MGH Harvard Medical School Advanced Clerkship in Clinical Neuroendocrinology. Her primary clinical focus is in pituitary disorders. She has extensive clinical experience in all aspects of pituitary care, particularly acromegaly, and has published clinical research papers in this area. She has received many awards for both research and teaching and clinical investigation. Today, she will be discussing the role of various T flavonoids on endocrine health and well-being. Our final speaker will be Professor Philip Devlin, who is the Chief of the Division of Brachytherapy in the Department of Radiation Oncology at the Dana-Farber Brigham and Women's Cancer Center, Harvard Medical School. Professor Devlin serves as the Executive Committee of both the American Society for Radiation Oncology and the Massachusetts Radiological Society. In 2008, he was named a Fellow of the American College of Radiology, and he is currently leading 2 funded studies as part of the ACR's quality research in radiation oncology. Today, he will be discussing the oxidative damage incurred during radiation therapy, and that could be greatly minimized in healthy tissues by the intake of T flavonoids. Thank you.
Hi everyone, I'm Siddhartha Jena, and today I'm going to talk a little bit about some of my visions towards expanding the toolkit for plant epigenome engineering. These are just some of my thoughts on what I believe the future of the plant biology, plant engineering, and sort of larger agricultural science field is moving into the future. So the problem as it sort of stands in the 21st century is that population growth has sort of necessitated more crops, more food to sustain the growing population. And this means faster growing plants, but also more nutritious plants, things that can deliver more sort of sustenance to a growing population. And simultaneously, the pressures brought on by climate change necessitate more resilient plants, things that can handle more extreme temperatures, for example, or drought, or even plants that sort of help us overcome some of the issues with the current state of the climate, such as plants that are better at fixing carbon. And just to drive this home, it's not as simple as sort of a physical presence for plants that the climate is changing so rapidly and in sort of unforeseen ways. This can not only have an effect on the sort of the temperature and the humidity and the moisture that a plant experiences, but also can lead to things like plants releasing different amounts of hormones, undergoing different immune responses, and being more or less susceptible to pathogens, which will, obviously deter from the health of the plant. So across sort of human civilization, we've used different forms of what I'll call traditional genetics to overcome agriculturally related problems. And that just involves breeding plants together to select for different traits, just as we have done for through animal husbandry and for a large, swath of different organisms throughout humans' existence. But this process is pretty iterative. That's evidenced by the fact that it's taken a long time to get certain features in fruits or vegetables. And more importantly, we may not be able to use traditional genetics to sort of converge on a solution for these sort of extreme situations like extreme heat, extreme cold, things like drought that we're facing today. And this is just showing, sort of the level of detail that's required, for example, in using traditional genetics to hand pollinate flowers. And robotics techniques and advanced machinery have allowed us to sort of scale these processes up to things like crop plants like maize, but it's still a bit limiting in the sense that it's combinatorially very hard to find that perfect genetic solution to a problem such as heat or cold resistance, for instance. But there is a possible future, which is this next generation of plant genome engineering. And I'm going to talk about sort of what this entails, as well as the potential and pitfalls of these approaches. So usually editing the genome these days involves either the CRISPR machinery or some sort of zinc finger-based delivery of a nuclease that's used to sort of mutagenize a target gene and cause that gene to lose function in a cell of interest, in this case, in a plant cell. And this involves delivering a guide RNA, which targets the gene of interest, as well as the Cas9 or CRISPR machinery, which is some kind of targeting protein, in this case a nuclease, that cuts the genome at that site. And then this causes often indels to appear, which can cause the gene to lose function. You then package those two components, in this case two components, together into the same plasmid, deliver it, as you can see, to a plant, and then screen for intended effects to see, exactly what kind of perturbations cause the result that you wanted. Delivery in plants is an active area of research, and it's a little bit different than delivering genetic products to animal cells or human cells, for example. Usually we use things like agrobacterium, which are just bacteria that carry that genetic cargo, like the Cas9 system. You then can inoculate plant cells with these bacteria and then cultivate them. You can even transplant them into full plants, and then select for your phenotype of interest. And recently, I haven't shown here, but viral vectors based on, for example, the tobacco rattle virus have been used to more efficiently deliver these genetic products to plant cells. But there's some limitations here, even though there's been a lot of advances in delivery and also what kinds of machinery we can deliver to plants for these genetic perturbations, there's a limitation on size. Any plasmid, whether it's a viral vector or pathogen of bacteria, has a certain carrying capacity, and there's only so many different things that you can put into these plasmids until they sort of run out of space, so to speak. And there's also a limit on the number of components. You can imagine saying, well, I have 4 different things that I want to introduce to these cells. Instead of putting them all in one plasmid, I could give the plant 4 different plasmids, but then you run into a sort of combinatorial wall where the chance of any one cell receiving all of these components that you've carefully picked becomes very, very low. And so ideally in the field of plant engineering and of cell engineering at large, we want to optimize for things that are as small as possible and that have as sort of few components as possible, which makes them very easy to deliver and then also very efficient in actually doing their job. And for plants, there's a sort of one more layer, which makes plant engineering, in my opinion, very interesting, but also a bit of a challenge, which is that although plants are sort of, for the most part, stuck where they are, they're rooted, and so they're spatially almost trapped, plants experience a huge amount of temporal variation. And so one example is the flowering of short day plants versus long day plants. Different plants or flowering plants have evolved to sort of respond to different day-night cycles in distinct ways. And that's why you get plants that flower during some types of light cycles versus not others. But what that means is that it may not be enough to cause a sort of permanent change in a plant, let's say by knocking out a gene using CRISPR. The better thing to do would be to somehow engineer a temporal response into a plant. So some kind of machinery that allows the plant to do a certain thing at a certain time when it has to, not just have that thing on or off permanently. And this is where I think engineering the epigenome could be of great use. So on the left is a sort of a very like granular image of a plant cell, and this is actually a plant sperm cell. So when plants need to reproduce, create more plants, this is of course important in growth of plants and the dissemination of things like crops. The genome, which is shown in light blue with that DAPI stain, changes its sort of condensation state. And you can see that normally you can see these sort of like round orbs of compact DNA in the row of images at the top. But when a particular protein called a histone is mutated, that DNA becomes more diffuse and it's not compacted nearly as well. And you can imagine that the sperm has to compact this DNA in order for the plant to effectively sort of reproduce in certain situations. But you can also see on the right that this protein, the single protein, which is very important in the case of this Arabidopsis plant for packaging its DNA in a very particular way, has actually diverged a ton throughout evolution of different plants. And so it raises the possibility that these proteins and other proteins in these epigenome, sort of these epigenomic protein families have heavily diversified throughout evolution to create particular types of behavior in plants that could then be, if we understood them a little bit better, ported from plant species to plant species. For example, a plant, to sort of make it more concrete, a plant that responds to certain environmental conditions by changing the way in which it reproduces could be engineered, that sort of mechanism could be engineered into a different plant that doesn't have that capability in order to make that latter plant better at sort of surviving in those environmental conditions. And as you can see from this evolutionary tree, there's tons of speciation in these proteins. This is actually a protein that's considered to not have evolved very much. As you can see sort of the extent of how much variety there is in even a very uniformly conserved protein relative to all the other proteins that can be found in the epigenome. So I think there's a huge amount of like sort of vast untapped potential in these chromatin-associated and epigenomic proteins that we have yet to sort of tap into for the sake of plant engineering. And so just to summarize what I've been talking about, I mean, just in a sort of diagrammatic sense, plants respond to environmental changes through a large variety of gene expression, and we can see this gene expression. We've been picking these mechanisms apart over many decades, are often the result of different types of interacting parts that turn on different genes or turn off other genes. Right now, our current toolkit is really just playing around with the toolbox that we are limited to, the toolbox of the plant in question. So we have the list of genes and things that we can do, for example, is to knock out that gene on the bottom, and we've sort of changed that plant response circuit by removing one of its components. But expanding this toolkit, by my definition, involves adding components. So, by looking through evolution and using some of these delivery methods and vectors that I discussed a little bit earlier in the talk, we can imagine altering this response circuit, not just by deleting certain parts of it, but by actually adding in new proteins that might do functions a lot more efficiently than existing proteins in that plant species, proteins that add new sort of regulatory nodes to these networks, like shown in the bottom middle. And then finally on the right, proteins that can cause, time varying things like oscillations that can help plants undergo better environmental feedback as sort of things like temperature, sunlight, and moisture change around them. So with that, I'd like to thank all of you for listening. I'd be happy to answer any specific questions over email or discuss this research program in more detail. This is where you can reach me, and for more information about some of my work, you can check out my website.
Good morning. My name is Rob Flamenhoff, and I am professor of medicine at Harvard Medical School and a chief of the Division of Hemostasis and Thrombosis at Beth Israel Deaconess Medical Center. And today I'm going to be talking to you about the cardiovascular benefits of tea. But before I do so, I thought I would go through some of the cardiovascular diseases that we'll be considering. So as a principle, the types of cardiovascular diseases that we'll be talking about in this talk are ones in which a clot occurs in the vasculature of a major organ. So if a clot occurs in your brain, that's a stroke. If a clot occurs in your heart, that's a heart attack or myocardial infarction. You can frequently get clots in the veins of your legs, and that's called a deep vein thrombosis. If it occurs in the arteries, then that's called peripheral arterial disease. You can also get clots in the vasculature of your lung. So here's a clot in your lung, and that's called a pulmonary embolism. And tea has been shown to benefit several of these conditions. So tea is associated with decreased stroke, with decreased heart attack, with decreased hypertension, and these have been attributed to antioxidant effects, also an improvement in lipid profile, which Dr. Noctegal will be speaking about after this talk, and improved glucose metabolism, which Dr. Noctegal will also address. So, the effects of tea in stroke are actually quite dramatic and have been shown in many studies now. So this is a meta-analysis that includes 9 studies and over 250,000 people. And the way to read these forest plots is that this line going down the middle is no effect. Over this area is benefit, and this area is worse outcomes. And so you can see with several different studies there's an average of a 36% decrease in stroke if one compares 1 to 3 cups of tea per day to less than 1 cup. If you drink more tea and you drink, let's say, up to over 4 cups and compare that to less than 1 cup, you can see there's actually a 72% decreased risk in stroke, which is quite substantial. The same trend is seen with heart attacks, although it's a little less dramatic. So if you drink 1 to 3 cups compared to less than 1 cup, then you can see there's approximately a 20% decrease in stroke. If you drink more than 4 cups compared to less than 1 cup, then there's a 32% decrease in stroke. So you can see there's a sort of concentration-dependent effect here. It seems that the more tea you drink, at least at these levels, the better off you are. Now, the cardiovascular effects of tea have been studied for a while now, and we have some understanding of what the constituents of tea that promote health are. And these include flavonoids, which are also found in fruits and berries. And these are rutin and isoquercetin I'll be mentioning. Also EGCG and ECG are rich in teas. And a sort of subclass of this are the galated polyphenols. And these, again, these include the EGCG, ECG, tannins, and theoflavins. And it kind of raises the question, what are flavonoids? So flavonoids are molecules that plants produce. They have this 3-ring skeleton, A, B, and C, and the plants have various enzymes that will produce different types of flavonoids. So the ones that are most abundant in tea are the flavin-thiols and flavanol. Isoflavones you may have heard of as being important in women's health. There are flavones and flavanones are also found in tea to a lesser extent. And the mechanism of the flavonoid benefit has also been studied, and it's clear that they have an antioxidant function. That is, they scavenge these free radicals that can contribute to vascular dysfunction and atherosclerosis. And this is just a schematic of that, where you have here quercetin, and you have a free radical, and the free radical will react with the quercetin, and that will neutralize the free radical. And then there's electron delocalization that keeps the free radical away from the vasculature. And although this scavenging property of the flavonoids is undoubtedly important, the newer thinking is that it's probably not the main benefit of flavonoids. And this is because your body has a lot of enzymes that are very powerful producers of free radicals, and other enzymes that are play a role in transferring the free radicals to proteins and nucleic acids where they cause damage. And so there's new thinking that the flavonoids may benefit by inhibiting these enzymes. And one enzyme that we have studied that the flavonoids appear to target is protein disulfide isomerase. Protein disulfide isomerase is a protein that is involved in protein folding when it's in your cells, but when it gets out of your cells, it can wreak all sorts of havoc. And it has this U-shaped structure where the catalytic domains, the domains that cause the problems, are in the A and the A prime subunits. And we found that PDI is a potent producer of thrombosis, that is, those blood clots in your blood vessels. It contributes to them in an important way, such that if we inhibit PDI, we can inhibit the formation of these clots. So we thought that it would be useful to develop inhibitors of PDI as antithrombotics. And so we performed a screen to evaluate, to identify novel inhibitors of PDI. And interestingly, our best hit was a molecule called rutin, which I just mentioned to you is a flavonoid. It has a sugar on the third position of the C ring. And we found that really any flavonoid that has a sugar in this position will inhibit PDI. And more recently, with collaborators in China, we have identified the exact position where the flavonoids seem to inhibit, and that is in this hydrophobic pocket. Here's a blow up, and we know to the atomic detail how this flavonoid actually inhibits PDI. But of course, what we're really interested is knowing whether these quercetin flavonoids block thrombosis. And for this, we used an in vivo thrombosis model. So this is a picture of the surgical preparation. This is in mice, and you can see we get a very clean preparation of a arterial here. And what we do is we hit that arterial with a laser, and we can see the accumulation of platelets in red and fibrin in green. So the platelets are the cellular component that causes clots, and the fibrin is the protein component that causes clots. So what happens if we infuse quercetin into the mice and then make these injuries? And you can see each of these has an injury, but you don't see any thrombus, particularly at the 0.5 mg per kilogram. And these are very low concentrations of isoquercetin that are able to inhibit thrombosis. In fact, we found that isoquercetin, which is very similar to rutin, inhibits thrombosis. It's very potently and is probably the most potent inhibitor that we've tested in this model. And so we've gone on to evaluate isoquercetin in patients. And so patients with cancer have a very high risk of developing thrombosis. So in collaboration with Jeff Zwicker, we evaluated 47 patients with advanced cancer. They received either 500 milligrams or 1,000 milligrams of isoquercetin. We obtained their blood at baseline, and after 56 days, we assessed for serum markers of coagulation, but we also evaluated clotting by ultrasound and kept track of bleeding. We found that the isoquercetin caused a 73% decrease in PDI activity, so we did appear to hit the target. And we also found a significant decrease in multiple markers of thrombosis, so there was a benefit at our primary endpoint that was significant. What was also, really interesting is when we did the ultrasounds, we found that there was no clotting at all. And this was not a randomized controlled study, so we didn't have a control arm to compare this to, but we could compare it to a historical control that had the same inclusion-exclusion criteria for enrollment. And in that study, there was something like a 20% or more rate of thrombosis. And so it at least appears that the isoquercetin is antithrombotic in these patients with cancer, and we're now moving on to do a randomized controlled study. Well, just as that data was coming out that we showed that there was a in patients that there was an effect of isoquercetin, the COVID pandemic broke out, and we were all sent home. We couldn't be in our labs were shut down, and we spent our time on the computer trying to figure out what we can do. This was particularly acute for me because there was it became very apparent quickly in the early stages of the pandemic that thrombosis was associated with COVID. So we wanted to come up with ways to combat COVID, but also combat thrombosis. And I noticed that there were a lot of docking studies. These are studies that, some modeling studies that people do on their computer showing that flavonoids inhibit SARS-CoV-2 main protease, the Mpro protein, which is involved in replication. And so I thought that perhaps it would be useful to identify flavonoids that both inhibited SARS-CoV-2 Mpro and therefore viral replication, and maybe they would also inhibit PDI and therefore thrombosis. And we have a very good high throughput lab at Harvard Medical School. And what we needed was a flavonoid library that was, a large library of flavonoids to test. So I looked all over the world for the largest commercially available flavonoid library that I could find. And lo and behold, it was a company in Wuhan that had a very large flavonoid library, over 1,000 flavonoids. And so I was in constant contact with folks in Wuhan, and using their flavonoids to try to find inhibitors of COVID. And we were able to find some. So this is actually the viral replication assay. You can see apigenin was very effective, but the most effective was PGHG. And so we evaluated whether PGHG would inhibit PDI activity as well. And it was a very potent inhibitor of PDI activity. And we found that these gallic acid moieties, which are known to have to be antioxidant, were necessary. There are 3 of these gallic acids on PGHD, and these were the necessary moieties to inhibit PDI. And so we looked, there are a lot of galatylated polyphenols, as I mentioned before, and we found that all of them inhibited PDI, and mostly with roughly the same potency. So we're now testing these in our animal model to see which ones are antithrombotic. And so far, PGHG is proven to be potently antithrombotic, and punicalagin, which is highly concentrated in pomegranates, is also antithrombotic. So, I've mentioned to you that these flavonoids and galatylated polyphenols are antithrombotic and therefore cardioprotective. Is there any way to enhance the concentrations in tea? So one idea is to use tea dust. So tea dust has a higher surface area to volume, and it enables more highly concentrated teas. More concentrated teas are going to have higher concentrations of flavonoids and galatylated polyphenols. And as I showed you, concentration matters because there's more cardiovascular benefit with more tea. Another possibility is to use blended teas or tea infusions. So natural phytochemicals in tea could be augmented with, for example, pomegranate extract, which is rich in punicalagin, or apples and citrus, which are rich in rutin. Another issue is that flavonoids are not, very well absorbed, and so one can enhance the absorption of flavonoids and therefore increase the delivery of these protective agents by mixing them with milk, ascorbic acid, black pepper. Special preparations have been tested using lysosomes, emulsions, and nanoparticles, and there can also be chemical modifications. Now, chemical modifications might not sound attractive because, here we are dealing with natural products and we don't want to, chemically modify them. But we can actually get around this by helping the plants make better flavonoids. So the metabolic pathways for these flavonoids are very well worked out and known in detail. And by slightly tweaking the, the enzymes within these pathways, we can make better flavonoids. So, for example, let's just say we have identified by our in vivo studies compounds that are galatylated and are antithrombotic. And so now we can engineer, by increasing the enzymes that gallylate the polyphenols, and we can make plants that make better higher concentrations of gallylated polyphenols. So one could envision a kind of pipeline where you identify the flavonoids that are beneficial and then engineer the plants to make those beneficial flavonoids. So just to summarize, I've shown you that tea consumption is cardioprotective for stroke, heart attack, and other cardiovascular diseases. Flavonoids and gallylated polyphenols that are found in tea inhibit enzymes such as PDI and thereby block thrombus formation in blood vessels. I've shown you that these flavonoids and gallylated polyphenols can mediate these antithrombotic effects in vivo studies, that the effects of tea are dose dependent, and we discussed some methods to increase flavonoid effects such as using tea dust, adding infusions of fruits and other consumables, enhancing the absorption, and generating tea plants that produce more cardioprotective flavonoids. And with that, I'd just like to thank all the people who have helped with these projects over the year, the funding sources, and here are my disclosures. And thank you very much.
Thank you for the introduction and for the invitation to speak today on flavonoids and neuroendocrine health. I'm Lisa Noctegal. I'm an associate professor of medicine and the clinical director of the Neuroendocrine and Pituitary Tumor Clinical Center at Mass General Hospital in Boston and Harvard Medical School. These are my conflicts. The potential neurocognitive and cardiometabolic benefits of tea are many, including protection from the development of dementia, protection from the development of Alzheimer's disease, decrease in hypertension, improved lipid profile, and improved glucose metabolism. Tea flavonoids have been associated with improved brain health, and large epidemiologic studies have shown a benefit of tea and flavonoids found in tea in several aspects of brain health, including improvement in cognitive performance with age, decreased risk of dementia, and decreased risk of Alzheimer's disease. In a community-based study among over 1,000 elderly people in Japan, tea intake was assessed via a survey, and cognitive function was tested by a Japanese version of a 30-point mini mental status exam. And what was found was that there was a dose-dependent decrease in the incidence of cognitive impairment. So you can see here on the slide that if you drink at least 2 cups per day in the black, you have a decreased risk of developing dementia or impaired cognitive function compared to the white, which is if you drink less than 3 cups per week. And here in the middle in the gray is moderate tea drinking of 4 to 6 cups per week, which didn't do as well as the higher quantity of tea with more than 2 cups per day. So this and many other epidemiologic studies really link not only tea, but the quantity of tea with a fair amount to the development of protection of cognitive impairment and dementia. And here you see similarly that if you take this study, which was again a very large study from the UK Biobank with over 100,000 participants followed over 9 years, that as you increase the amount of flavonoid-rich food you consume, you decrease your risk of developing cognitive impairment. And if you look specifically at subpopulations, it was interesting to learn that there was even more risk reduction in people that had already a high genetic risk of dementia, had hypertension, or had depressive symptoms. And these people were even more protected by the effects of tea. Here you see we have spoken a little bit about dementia, and in a talk before this, you heard about the antioxidant, vascular function, and platelet aggregation benefits of flavonoids. And for the rest of this talk, I am going to tell you about an area that is closest to my field of endocrinology, and that's diabetes, hypertension, hyperlipidemia, and inflammation. So, does tea help prevent diabetes? There's a little bit of a mixture of responses to this in the literature. But what I show you here is a meta-analysis of 7 studies that estimated the association between tea and the development of type 2 diabetes. And here you can see that's on this side of the line are all the studies that showed a decrease in the incidence of developing type 2 diabetes. And on this side of the line is the studies that showed an increase in the development of diabetes. And you can see that some of the studies kind of cross the line and really show there wasn't a big difference. But taken together, if you average them all out, you see this cluster, showing that there probably is a true risk reduction of the development of diabetes associated with tea consumption. And maybe some of the reasons for the differences in these studies are the actual designs, where different studies had different tea intakes and other different aspects to their designs. What about blood pressure? In a study of over 15,000 participants in the USA, higher total flavonoid intake was associated with lower blood pressure and a lower risk of hypertension. This was specifically related to flavin-3-ols, which are high in tea and tea dust, as well as other flavonoid components that were linearly and negatively related to the presence of hypertension. Studies have shown that both tart cherry juice and berries were associated with a decrease in blood pressure, and there is a little bit of evidence that angiotensin-converting enzyme may be inhibited by the presence of compounds in fruits and tea, and that may be the mechanism for the blood pressure lowering. And I should point out, because many of you listening may be on medications that actually work on this enzyme, which you may know as an ACE inhibitor. Many Americans and other people around the world will take ACE inhibitors to treat high blood pressure. You may know them as lisinopril and losartan. There are so many of them. But it turns out that these compounds may actually have an effect on that same signal pathway. What about cholesterol? Regular tea consumption significantly reduces total cholesterol, LDL, triglycerides, and they increase HDL. And just to remind you, HDL is the good guy and LDL is the bad guy. So lowering LDL is very helpful for protecting heart disease, and increasing HDL is very helpful for protecting heart disease. And that's exactly what flavonoids in regular tea consumption does. And it seems that there's an antioxidant effect, as you have heard in the prior talk, but that specifically there's an inhibition of the oxidation of lipoproteins. And it turns out that if you oxidize LDL, it's even worse. And so preventing its oxidation really helps prevent the atherosclerosis caused by elevated levels and oxidized LDLs. Flavonoid-rich diet is also associated with a lower risk of fatty liver disease. So when you see NAFLD, it is non-alcoholic fatty liver disease. And in this study, 121,064 adults were followed over 10 years, and those that had the highest quartile of flavonoid score had the lowest incidence of fatty liver. And one strength of this study was they actually had MRIs that showed the decrease in the liver fat. High intakes of apple and tea were associated with a lower rate of fatty liver. And I know a lot of you are listening to this in India, where you have plenty of tea. And I am talking to you from Boston, where at this moment in the fall, we have plenty of apples ripe on the tree. So together, we should be very effective in helping prevent liver disease. So it turns out that tea inhibits 11-beta-hydroxysteroid dehydrogenase type 1, which is an enzyme that is important in activating cortisol. So inhibiting this enzyme effectively lowers cortisol. That's interesting because stress causes very high cortisol levels. And very high cortisol levels, as you may know, is disruptive for blood pressure, causes heart disease, hyperlipidemia, and is associated with diabetes. And so lowering cortisol may be a very important way of reducing metabolic syndrome and some of these other cardiometabolic consequences. And that may be how some of the flavonoids are working. You can see here in this picture that this little blue molecule is actually a flavonoid from tea, and it fits very nicely in this spot, as you can see, within this scaffold, which is the protein that is that enzyme. It's 11-beta-hydroxysteroid dehydrogenase. So if there's a little inhibition here of that enzyme, you get a lowering of cortisol, and we see that here. A lot to swallow, but what I'm trying to excite you with is the idea that one of the mechanisms here from all these amazing things that we've seen that flavonoids can do is they may actually inadvertently be decreasing cortisol. And there was one study that looked at this and actually showed that when you give an ingestion of polyphenol-rich dark chocolate, 500 milligrams per day over 4 weeks, you're seeing lower cortisol levels compared to controls that had dark chocolate without that flavonoid component. Finally, I'll leave you with an interesting thought, which is, I think in the busy times that we live in, we all have those times where we have to suffer from some sleep deprivation. And it's shown actually that in humans, there's cardiometabolic effects of sleep deprivation, and that antioxidants in tea may alleviate this. And there was an interesting study in rats where they gave tea and demonstrated a protective role restoring antioxidant enzyme levels and suppressing lipid peroxidation and improving nitric oxide levels in rats with chronic sleep deprivation. So the reason that this slide is so interesting to me is that think about what happens when you don't get enough sleep and how the first thing you want to do when you are so tired is grab a cup of tea or coffee or something with caffeine in it. And so it makes sense that we're craving something that is actually working physiologically to help aid some of the injury that is happening at the vascular and cardiac levels in relation to the stress of sleep deprivation. So just to summarize, there have been many favorable effects of dietary flavonoids that are seen on blood pressure, risk of diabetes, cholesterol, and brain health. And high flavonoid-containing tea, apples, berries, dark chocolate, and many other foods may actually contribute to health and wellness. Their antioxidant properties maybe decrease in ACE levels, angiotensin converting enzyme levels, and possibly a reduction of cortisol may play a role in the mechanisms of their promotion of health. Overall, as we conclude this section, what should we tell our patients? What do we tell our patients? Well, I can tell you that at the Memory Center at Mass General, they do give patients a very helpful guide to diet, where there is a kind of a Mediterranean diet that's an evidence-based suggestion for brain health, really focused on high flavonoid foods like berries and apples and fruits. And more studies are needed in terms of what we know and can suggest in terms of treating and avoiding metabolic syndrome, diabetes, and weight loss. But for now, it seems, judging the data we have to date, and we need more, that tea drinking may really help.
Hello, I am Dr. Philip Devlin. I am a radiation oncologist at the Brigham and Women's and the Dana-Farber Cancer Institute in Boston, Massachusetts. I'm deeply honored at the invitation to spend some time with you and to teach you about radiation energy as a way to cure cancer. I understand this is an audience of a very broad-based audience, and I'm going to invite you to enjoy a little bit of science with us just now. But first, the disclosures that I have 2 textbooks out there and I get royalties. I'm on the Scientific Advisory Board of Porosome, and I may discuss off-use label of medical devices. So let's get dug right in. We've got some learning goals in just this short time to learn about radiation as just energy, that energy can be transformed, this energy transformation can break the bonds of water, that radiation kills by this indirect effect, and this indirect effect is called oxidation. Normal, innocent bystander cells are affected less by oxidation and also need antioxidation to protect so they can repair. Now let's start and dive right in with a real clinical model. This is an ugly picture. This is a rare skin cancer that's just been treated with radiation therapy. You can see right away there are 2 different colors. There are 2 black spots marked by the blue arrows, and then the rest of the foot looks like it's got a terrible rash and little white spots which happen to be skin regeneration. The name of this reaction to radiation is called moist desquamation, just to know where we are. So how come, if the whole of this area got the same radiation, that some of it looks black, which is dead, and others of it looks red, which is inflamed but healing? The difference between how radiation works on cancer and how radiation does work on normal tissue, but to a lesser extent. This is from the United States Nuclear Regulatory Commission that reminds us that radiation causes ionizations of atoms. That's the breaking of atoms in part. It can affect molecules, which can affect cells, which can affect tissues, which can affect organs, which can affect the whole body. And I'm going to add one to their list for you just now. See, they're showing you that this can go everywhere with this very neat diagram that radiation can affect, for good or for bad, every part of our body. My favorite teacher at Memorial Sloan Kettering, Dr. Murray Brennan, taught me this. Philip, the patient wants you to save my life, to save my organs and limbs, and here's our addition, to save my function. Radiation targets cancer cells to damage DNA, leading to cell death. Radiation splits the water molecule H2O into ions, the H plus and the OH minus. Both of these ions are hungry for an electron to fill their electron shell. Now, it's very important to note that the backbone of DNA are held together with relatively weak electron bonds. And so that hungry ion is able to steal electrons from the DNA backbone. The DNA falls apart. If we break both strands, this equals cell death. This is what we want to happen to the cancer cell. Stealing electrons is called oxidation. The ions are like free radicals that destroy DNA bonds. So what is the role then of antioxidation in normal tissue protection? The way to look at it is antioxidants are like the vacuum cleaners of the nucleus of the cell to scoop up ions and free radicals before they can break the DNA of normal, innocent bystander cells. Some people call some of these antioxidants scavengers. It's a good word. They're hoovering up the bad guys. Okay, good. So the question then is, why doesn't radiation kill bystander cells the same way it kills cancer cells? Well, cancer cells are always duplicating. The DNA unfolds the coils. When they're unfolded, they are in water. This is a hydrophilic environment, and therefore the energy of the radiation can make lots of ions. So we can hurt the cancer cell, but normal tissue are normally not duplicating, and the DNA is folded and the coils are closed. This is a very hydrophobic environment with very little ionization damage. When cells, normal cells, are well nourished, there are lots of antioxidants. Anions are more likely to be neutralized. If there is a strand break, just one of the DNA strands would break, and DNA can repair itself if only one strand is broken. So let's just look at this again. So I only showed you half the story at the beginning, that there is this foot really with two pictures. One, that the radiation has killed the cancer, and two, that the normal tissue has had some injury and affront to it, but the picture next to it is 6 weeks later, the same foot, same person, with a complete resolution of the normal tissue and a complete disappearance of the tumors. So that is the good news about radiation being able to kill preferentially cancer cells and hopefully not harm normal tissue too much, so that we can return to save the life, save the limb, and as I love to say, save the function. So save the function for this man. This man came in having been offered a foot amputation by his doctor. That wasn't acceptable. When we took this photograph 6 weeks after this radiation, he had completed a 6-mile hike on Cape Cod. It was such a good, his function was fully restored. So this is the good news of radiation. While talking to my friends in Mumbai, you cannot not mention the wonderful contribution of the Tata Memorial Hospital in the development of image guidance to cure another cancer. This is an image or two images of cervix cancer brachytherapy. What we see in the right-hand picture is the orange area is the cancer. And what we see to the right is that the yellow area is the bladder and the pink area to the back is the rectum. And so we're able to use image guidance to direct the radiation fully to where it needs to be and optimally spare normal tissues from being damaged. Thus, this disease, this is cervix cancer caused by human papillomavirus, which is spread in young people and would lead to death if we were not able to be so vigorous with the radiation and chemotherapy so as to save these young women. And the Tata Memorial Hospital, right there with you all, is a world leader under the direction of Dr. Umesh Mantashetty. So if you're in the audience, greetings. So again, we've achieved our goals. Radiation is just energy. Energy can be transformed. It's transformed by breaking the bonds of water. Radiation kills cancer by an indirect effect. This indirect effect is oxidation, stealing electrons from the DNA backbone. Normal, innocent bystander cells are affected less, but need antioxidation to protect and so can repair by scavenging these ions. I just want to say thank you to the organizers for inviting me to give this talk. I will remain available through the chat for questions. Thank you very much.
I would like to thank Professor Banu P. Jena for organizing this international symposium. We've been exposed to really cutting-edge research and technology from the top scientists at Harvard Medical School and Harvard University. It's been really informative, and I think there are a lot of learnings on what T flavonoids can do for health, and also how global health can improve by increasing the consumption of tea through tea molecules which really work on specific diseases. I would like to thank Dr. Siddhartha G. Jena for his talk given on engineered plants for nitrogen and carbon capture. I would also like to thank Professor Robert Flomenoff for his talk on flavonoids and cardiovascular health. And I would like to thank Professor Lisa Nottingale for the talk on flavonoids and neuroendocrine health. And finally, thank Dr. Professor Philip M. Devlin for his talk on radiation, a natural and anti-cancer molecule of flavonoids. I will now request Professor Jena to give the concluding remarks.
Thank you very much. Thank you very much, Mr. Matthews, and thank you all in the audience and online attending this symposium. It's a true privilege actually to be able to do this with KDHP, because KDHP has been engaged for quite a while in trying to not only diversify the use of tea globally, but also bring its benefits to humanity, not only in India but worldwide. And this organized symposium was jointly held through the discussion and the progress that has been made with the joint venture with KDHP and Nytroponix. You have been able to just skim the surface of what we have achieved, because it's both these KDHP and Nytroponix have patent rights on many of these things. You just heard the very surface of what tea can bring, what benefits that can be brought. But there is tremendous progress already been made, where a launching of infused tea, which is highly, highly effective compared to what you just heard. And this is going to help society. India, with its national drink, this tea, would be proud of this, and I'm sure that you will join me in this endeavor to make this joint venture not only successful, but globally impactful. So with that, I want to thank you very much for attending.